This is really a great review article, and then you just click this and it would get that journal. It's not a very long journal either. Okay, so Dr. Jen already mentioned the main area in the brain that we think that is not quite right is the prefrontal cortex. And the frontal cortex really takes about half of the front brain, and then the prefrontal cortex is maybe 2/3 of the frontal cortex, the front part of it. And this area is made mainly for decision making, emotion, and decision making is mainly for the delay gratification type of decision. Of course, attention too. So you can see that if something is not quite right with that, then you tend to go off tangents and go off doing things that are really not your primary goal at this point. So there's a lot of evidence suggesting that the PFC, the prefrontal cortex, is a key area. And the two major neurotransmitters that are involved in ADHD are believed to be norepinephrine and dopamine. You immediately realize that dopamine is involved in so many things. You can almost say that, oh, dopamine is involved in everything. Therefore, hopefully you do not try to aim at dopamine, so because now you will mess up a lot of things. So norepinephrine, there are some generalization, you get a sense that generalization, there's always exceptions, but the generalization helps you to understand the drug's mechanisms. So stimulation of the alpha, alpha 2A receptor would improve ADHD symptoms. Hence, one of the drug classes is alpha 2 agonist. And then there are also evidence suggesting that actually stimulating alpha 1 is bad, stimulating beta in the brain is bad also, okay? So we try to avoid those drugs, so there are no such drugs on the market for these receptors to help ADHD. And then dopamine, we do not truly know what receptor is critical for dopamine's effect, but also there is a bell-shaped dose response curve for dopamine, which means that if there's not enough dopamine, you might have problem. If there's too much dopamine, you might have problems. So there is a bell-shaped curve that you need to be the perfect concentration. And so this is simply to show you that norepinephrine, the main cell bundle of cells are in the locus ceruleus and then sent to the prefrontal cortex. In this slide, it suggests that norepinephrine's one of the key functions is dampens the noise. If you think about the noise signal ratio is the key mental scaffold to think about ADHD. So if you can dampen the noise, you can help to focus on the important thing. And then dopamine, the main cell that generates dopamine is in the ventral tegmental area, so they send out to that. And then one concept you can think about is dopamine may enhance the signal. So enhancing signal, suppressing noise, that's how you can improve the ADHD's behavior. I also want to emphasize the bell-shaped curve concept. So this is the prefrontal cortex abilities in decision making, delayed gratification, attention, etc. This is the amount of norepinephrine or particularly dopamine. If there's not enough norepinephrine and dopamine, then you are lack of energy. You are fatigued, lethargic. But there's too much, then you feel really agitated, excessive stress. So you need a perfect concentration or activity of dopamine and norepinephrine in the brain to have a sufficient alertness and without being distracted. So that is the general concept of how to manage ADHD. But in fact, it's very difficult to really do any of these studies in brain. So we have only 3 major drug classes. The first one is at the pharmacological mechanism level is norepinephrine and dopamine reuptake inhibitors, NDRIs. But all of this on the top two groups, they are also called stimulants, CNS stimulants, because they do stimulate the brain. The last one, bupropion, you learn it in antidepressant, right? It is a norepinephrine dopamine reuptake inhibitor, but it is generally not classified as stimulants. Bupropion is not, you do not call bupropion as stimulant. It is simply NDRI, okay? Only the methylphenidate or Ritalin and amphetamine analogs, they are called CNS stimulants. And then the next group that Dr. Jen already mentioned is actually losing people don't popularity is atomoxetine. This is a selective norepi reuptake inhibitor, so it doesn't affect dopamine reuptake as much, so it's focused on norepi. And then the last class are the alpha-2 agonists, because alpha-2 receptor stimulation can improve ADHD symptoms. And there are only 2 drugs that are used for that. And guanfacine is the more popular. So this is the few slides that talks about MedChem. So this is amphetamine, structure of amphetamine. If this chiral center is the dextro isomer, then it's called dextroamphetamine. And the so-called amphetamine pill would contain both the D and the L, so it's a mixture of both isomers. So you can immediately get a sense that this really look like dopamine and norepinephrine, this drug. If you take away this methyl group, take away, and then add OH on this carbon, and then another OH to this carbon, then it's dopamine, right? And then if you add another OH on this, on the dopamine structure, then it becomes norepinephrine. So then amphetamine's structure is very similar to both dopamine and norepinephrine. So you can imagine that they can interact with the transporters and receptors and even enzymes that would affect dopamine and norepinephrine's effect in the brain. So that's how amphetamine works. So because its structure is very, very similar to dopamine and norepinephrine. And then methylphenidate is, you can also just squint your eye, you see the structure are the same, and then this is the same nitrogen, and there is a carbon, so you just modify that. And again, it's the dextro form of methylphenidate is the more potent, more active form. And then methylphenidate as a drug is a racemate of both the D and the L form. Dr. Jain mentioned this dexamphetamine. It is gaining popularity. It's really just amphetamine plus a lysine on the tail of that. And this is the amino acid lysine. So this compound by itself is not pharmacologically active. So it is a prodrug. It requires to be cleaved to this area, this bond, and then to produce amphetamine to be effective. So, so it is a prodrug, and the good thing is that you have a more smooth dose blood concentration. So therefore, you don't have the peak and trough effect of some of these earlier drugs. Okay. In order to speak more about how this drug works, so I just want to review again how the norepinephrine or dopaminergic nervous system and how they were made and released. So dopamine can be reuptake into the neuron, or it can be made from tyrosine via dopa and then make into dopamine. And then dopamine would be taken up by this GAT gene, right? The VMAT transporter that into a secretory vesicle. And inside the secretory vesicle, if it has the right enzyme, then it can be turned into norepinephrine. So depending on whether the existence of this enzyme, this could be a dopaminergic neuron or a norepinephrine neuron, right? So either one of these would be in the secretory vesicles. So when an action potential comes along, this secretory vesicle would merge with the presynaptic membrane, release either dopamine or norepinephrine. It would interact with the receptors, and then some of them would be reuptaken by a transporter. And this transporter can be a dopamine transporter or norepinephrine transporter, depending on the nerve ending. And then some of that would be degraded by the MAO, monoamine oxidase. The ones that is not degraded then can be reuptaken into the vesicle to be reused, right? So that's the whole story. And how do the stimulants, this is the methylphenidates, the amphetamines, work? They work primarily by inhibiting the reuptake of dopamine and norepinephrine. Okay, so in this graph, the yellowish, greenish bar represent amphetamine, and then this greenish thing represent norepinephrine, and then the grayish thing represent dopamine. So amphetamine can block the reuptake of norepinephrine back into the nerve ending, as well as dopamine. Okay, the general idea is that they may work better in blocking norepinephrine than dopamine, but that's animal data. Whether it translates to human is not quite clear. So that's the primary mechanism of action. But in addition to this primary mechanism, there's also additional mechanisms that have been shown for the CNS stimulants. So they can actually increase the release of dopamine or norepinephrine into the synapse. They can also inhibit monoamine oxidase in the nerve endings, and they can also directly interact with the receptors and stimulate them. Okay, so all these effects, all these functions seem to enhance both the noradrenergic and the dopaminergic activities in the brain. But conceptually, this is the main function. This is the primary mechanism of action. So there are rat studies. If you give methylphenidate or Ritalin in the rat, and you measure norepinephrine or dopamine levels in different parts of the brain, you can see that this is a subcortical region, which means that you take away the cortex and the rest of grind up the rest of the brain. And then if the placebo defines 100%, you can see that both norepinephrine and dopamine will increase maybe 30% or so in the rat. But then if you only look at the prefrontal cortex area, you can see that norepinephrine increased by 300%, and then dopamine increased by 200%. So all these are much dramatic effects. So hence, it is believed that CNS stimulants, methylphenidate and amphetamines, are primarily working on that, but we cannot rule out the possibility that it's also working on other parts of the brain also. So let's talk about the drugs per se, and then their formulations. At the end of that, we'll have one summary slide to talk about the adverse effects of all of the CNS stimulants. So methylphenidate, we'll start with the short-acting methylphenidate formulation. So you are all familiar with Ritalin, the very short-acting tablet, and then there are also chewable tablets, liquids, and then also dexmethylphenidate, which is the more active form. It's a tablet, Focalin. So these short-acting formulations, the onset is 30 minutes to an hour, so very quickly come out. And then but the duration of action is in 3 to 5 hours, which is not very good to last a full school day for a child, right? But it has advantages and disadvantages. The advantages is that they probably are very useful as a top-up. So at the end of the day, when the child is given a intermediate acting pill, at the end of the day, the pill is the effect is going away. Maybe this is a good time to give a short